So dear students, concerned with this biotechnology, initially you have come across three different definitions of biotechnology. Initially, you have come across three different definitions of biotechnology. Followed by that, I introduced principles of biotechnology. One is genetic engineering, second is bioprocess engineering. Followed by that, I introduced the tools of recombinant DNA technology. Under that, the one which I covered is enzymes. Under enzymes, we have come across two types. One is commonly called as molecular scissor. Another one is called as molecular stitchers. Actually, I am talking with respect to restricted endonucleases and DNA ligase. Restriction, endonucleases and DNA ligase. Followed by that, even if I come across the nomenclature of restriction endonucleases. So today, I plan to introduce one more tool of recombinant DNA technology. One more tool of recombinant DNA technology. That is nothing but cloning vectors. Cloning vectors. Cloning vector. First question arises, what is this cloning and what is the meaning of vector? Cloning is the making multiple copies, making multiple identical copies of any template DNA. Look over here. So second, today I plan to introduce one more tool of recombinant DNA technology, cloning vector. Cloning vector. First a question arises, what is this cloning? Then I will discuss vector. Cloning means making multiple identical copies, of any template DNA. You know DNA, deoxyribose nucleic acid. It is commonly called as master molecule in the biological system because it will control all the activity of the cell directly or indirectly. DNA is commonly called as master molecule in the biological system because it will control all the activity of the cell directly or indirectly. Template means master copy, or you can call it as a blueprint. Okay, cloning means making multiple identical copies of any template DNA. Vectors means they will carry desired genes. So common meaning of vector is a carrier. As you have come across mosquitoes. Okay, mosquitoes are the carrier of malarial diseases. Same way here, vector means they will carry the desired genes. They are the carriers of desired DNA. Vectors are the carriers of desired DNA. Under that, many examples are there, but for your syllabus, they are quoted two examples. Vectors are the carriers of desired DNA. Desired means unique DNA or special DNA or the DNA which is having some identical characters. Okay, vectors are the carriers of desired DNA. Under that, for your syllabus, they are quoted two examples. Okay, one is bacteriophages, and another one is plasmids. Bacteriophages and plasmids. Already we have come across bacteriophages. Viruses are classified based on different criteria. Viruses are classified based on different criteria. One is based on the type of the cell which they infect. Initially I covered viruses which infect the bacteria are called as bacterial viruses. Viruses which infect the bacteria are called as bacterial viruses or bacteriophages. Bacteriophages, because of their high number per cells, have several copies of their genome in bacterial cell. Look over here. Two examples for the cloning vector is bacteriophages and plasmids. Viruses which infect the bacteria are called as bacterial viruses. Viruses which infect the bacteria are called as bacterial viruses. Because of their high number per cell, have several copies of their genome in a bacterial cell. They are having several copies of genome in a bacterial cell. Now question arises, what is this genome? Total number of genes located in a haploid set of chromosome is called as genome. Total number of genes located in a haploid set of chromosome is called as genome. Look over the definition. Total number of genes located in a haploid set of chromosome is called as genome. I hope that the point is clear. Viruses which infect the bacteria are called as bacterial viruses or bacteriophages. Because of their high number per cell, have several copies of genome in a bacterial cells. Because of their high number per cell, have several copies of genome in bacterial cell. Total number of genes located in a haploid set of chromosome is called as genome. Now look over, the next matter is related to plasmids. Plasmids are the extra chromosomal self-replicating circular double-stranded extrachromosomal self-replicating circular closed double-stranded DNA found in the cytoplasm of bacterial cell. Look over here. When they will ask what is plasmid, you have to write this complete meaning. Plasmids are the extra chromosomal. You know already, chromosomes are the nuclear component having specific organization, individuality, structure and function. Otherwise, back to PUC first year, chapter cell, the unit of life, where you come across the meaning of chromosome. Chromosomes are the nuclear content of the nucleus having specific organization, individuality, structure and function. The chromosomes are the thread-like structure present inside the nucleus. Chromosomes are the thread-like structure present inside the nucleus. Extrachromosomal, self-replicating. So back to molecular basis of inheritance. Now move to molecular basis of inheritance, where we come across meaning of replication. Autocatalytic function of DNA. One of the most important property of the DNA is that important property of the DNA is that it will manufacture an exact copy of itself. So this process is called as replication. One of the most important property of the DNA is that it will manufacture an exact copy of itself. So this process is called as replication, or the process of formation of replica is known as replication. Replica is nothing but identical copies or duplicated copies. The process of formation of replica is known as replication. Replica is nothing but identical copies or duplicated copies. So self-replicating, circular, as you have come across already the picture of PBr322, circular, it is round in shape, closed, completely packed, double stranded, already we have come across double helix model of DNA proposed by Watson and Crick. Double helix model of DNA proposed by Watson and Crick. So look over, plasmids are the extra chromosomal, self-replicating, circular, closed, double-stranded DNA. Actually, they are present slightly away from the normal chromosome. Therefore, they are called as extra chromosomal or accessory chromosomes. So, plasmids are present slightly away from the normal chromosome. Therefore, they are called as extra chromosomal or accessory chromosomes. But please remember, they are capable of undergoing self-replication independent of the host DNA. Please remember this line. They are capable of undergoing self-replication independent of the host DNA. They are capable of undergoing self-replication independent of the host DNA. Actually, they are present in the cytoplasm of bacterial cells. They are present in the cytoplasm of bacterial cell. You know already cytoplasm. Protoplasm is equal to cytoplasm plus nucleoplasm. Protoplasm is equal to cytoplasm plus nucleoplasm. Cytoplasm is equal to protoplasm minus nucleoplasm. Protoplasm is equal to cytoplasm plus nucleoplasm. Cytoplasm is equal to protoplasm minus nucleoplasm. So I hope that the definition is very clear. Okay. So plasmids are the extra chromosomal, self-replicating, circular, closed, double-stranded DNA present in the cytoplasm of bacterial cells. They are also called as extra chromosomals or accessory chromosomes. Okay, accessory chromosomes. But I requested to remember, they are capable of undergoing self-replication independent of the host DNA. They are capable of undergoing self-replication independent of the host DNA. So many examples are there for the plasmids. So I put forward two examples for the vectors. One is bacteriophages, second plasmids. Even cosmid is there, lambda phages are there, many more examples are there. Under plasmids, two examples you can easily notice in any textbooks. It is PUC-18 and PBr322. PUC-18 and PBr322. Plasmid of University of California number 18. Plasmid of University of California number 18. Plasmid of Bolivar and Rodriguez strain number 322. Look over the expansion. Plasmid of University of California number 18. It is so named because it was developed by a team of scientists belongs to University of California. Plasmid of University of California is so named because it was first developed in the University of California. Another example is plasmid of Bolivar and Rodriguez strain number 322. plasmid of Bolivar and Rodriguez, strain number 322. So plasmid of Bolivar and Rodriguez, strain number 322. You are going to study in detail. Plasmid should have origin of replication site. It should have ORI, origin of replication site. Second, selectable marker. Third is cloning site. So look over here, dear students. Plasmid should have origin of replication, selectable marker and cloning site. It should have origin of replication, selectable marker and cloning site. Now first look over origin of replication. This is the sequence of nucleotide. This is the sequence of nucleotide from where the process of replication starts. Origin of replication is the site, or it is the sequence of nucleotide. It is the sequence of nucleotide from where the process of replication starts. Already I covered the definition of replication. The process of formation of replica is known as replication. It is one of the most important property of DNA, or it is an autocatalytic function of DNA. It is the sequence of nucleotide from where the process of replication starts. You know already nucleotide. Nucleotide is equal to nucleosides. Nucleotide is equal to nucleosides plus phosphates. Okay. Nucleotide is equal to nucleosides plus phosphate. Nucleoside is equal to nitrogenous bases plus pentose sugar. Okay. Now look over two more related points with respect to origin of replication. Okay. One, it is the sequence of nucleotide from where the process of replication starts. It is the sequence of nucleotide from where the process of replication starts. When the foreign DNA or when a piece of DNA, when alien DNA or foreign DNA is ligated to ORI or origin of replication site, it is made to replicate within the host cell. When the foreign DNA is ligated to origin of replication site, it is made to replicate within the host cell. Even this origin of replication site will decide the number of copies. Origin of replication will decide even copy numbers. When you are planning to get several copies of required or desired DNA, when you are planning to obtain or produce several copies of desired DNA, you have to select such an vector which will facilitate high copy numbers. Look over here, the PBR 322 or it may be an PUC-18 or plasmid should have origin of replication. This is the sequence of nucleotide. Origin of replication is the sequence of nucleotide from where the process of replication starts. When the foreign DNA is introduced into the origin of replication site, it is made to replicate within the host cell. Even it will control the copy numbers. Origin of replication will control even copy numbers. When you are planning to get several copies of a desired DNA or foreign DNA, you have to select such an vector which will facilitate high copy numbers. Now look over the second matter, selectable marker. It will eliminate non-recombinants. It eliminates non-recombinants. It will eliminate non-recombinant and selectively permitting the growth of recombinants. selectable marker, eliminate, non-recombinant. Instead of that, you can also call it as a non-transformant. Selectable marker, eliminate, non-recombinant, and selectively permitting the growth of recombinant. Non-recombinant, or you can call it as a non-transformant. Recombinant, transformant. Look over here, take care of these points. Selectable marker, eliminate, non-transformant or non-recombinant, and selectively permitting the growth of transformant or recombinant. I will introduce two technologies, how they are going to identify recombinants from non-recombinants or transformants from non-transformants. Two simple techniques which helps to identify the recombinant from non-recombinant. Followed by that, I am going to introduce cloning site. Okay, now take care of this matter. First point, when the foreign DNA is ligated to, when the foreign DNA is ligated to foreign DNA, or instead of that, you can call it as a recombinant DNA, is introduced into, when the foreign DNA is introduced into or recombinant DNA is ligated to beta galactosidase, when the foreign DNA or recombinant DNA is ligated to beta galactosidase, it will loses activity. This is called as insertional inactivation. When the recombinant DNA is linked to beta galactosidase, the enzyme will loses activity. This process is called as insertional inactivation. You know already this enzyme. Any word end with suffix ase is the name of the enzyme. Any word end with suffix ase is the name of the enzyme. This beta galactosidase we have come across in Lacoperon concept. Back to molecular basis of inheritance and try to recollect this enzyme. In Lacoperon concept, you have come across structural genes, operator gene, promoter gene, regulator gene, repressor gene, and inducer gene. In Lacoperon concept, we have come across structural genes, operator gene, promoter gene, regulator gene, repressor gene, and inducer gene. There are three structural genes are there, Z, Y, A. Z will code for beta galactosidase, Y will code for permease, A will code for trans-acetylase. I hope that you are recalled this matter. Beta galactosidase. There are three structural genes are there, Z, Y, A. Z will code for beta galactosidase, Y will code for permease, A will code for trans-acetylase. When the recombinant DNA is linked to beta galactosidase, the enzyme will become inactive. This process is called as insertional inactivation. As a result, chromogenic substance as a result, chromogenic substrate is not expressed in the colony, and colony will appear colorless, or you can call it as a white. Look over here. When the recombinant DNA is connected to beta galactosidase, the enzyme will become inactive. This process is called as insertional inactivation. As a result, chromogenic substance is not expressed in the colony, and colony will appear colorless, or you can call it as a white. Okay, so when the foreign DNA is not ligated to, beta galactosidase, the enzyme is active. As a result, chromogenic substance is expressed in the colony. Chromogenic substance is expressed in the colony, and colony will appear blue in color. When it appears blue color, then this clear indicates that it is non-recombinant. Okay, look over here, take care of this matter because it will make you under confusion. When the recombinant DNA is ligated to beta galactosidase, the enzyme will become inactive. As a result, chromogenic substance is not expressed in the colony, and colony will appear white in color. When the recombinant DNA or foreign DNA is not ligated to beta galactosidase, enzyme is active, and chromogenic substance is expressed in the colony, and colony will appear blue color. When it appears blue color, this indicates that they are non-recombinant. When it appears white color, then it is recombinant. Okay, ಸ್ವಲ್ಪ ಇಲ್ಲಿ ನೋಡ್ಕೊಳ್ಳುವಂಥ ವಿಷಯ, ಯಾವಾಗ ನೀವು ಫಾರಿನ್ ಡಿ ಎನ್ ಎನ ತಂದು ಬೀಟಾ ಗೆಲೆಕ್ಟೋಸೈಡೇಸಿಗೆ ಹಚ್ತಿರೋ ಅವಾಗ ಅದು ಎನ್ಸೈಮ್ ಏನಾಗುತ್ತೆ, ಇನ್ಆಕ್ಟಿವ್ ಆಗಿ ಬಿಡುತ್ತೆ. ಅದರಲ್ಲಿ ಇದ್ದಂಥ ಕ್ರೋಮೋಜೆನಿಕ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಏನಾಗುತ್ತೆ, ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಲ್ಲ. ಹಾಗಾಗಿ ಇಡೀ ಕಾಲನಿ ವೈಟ್ ಕಲರ್ ಕಾಣುತ್ತೆ. ಯಾವಾಗ ರಿಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎನ ನೀವು ಬೀಟಾ ಗೆಲೆಕ್ಟೋಸೈಡೇಸಿಗೆ ತಂದು ಹಚ್ಚಲ್ಲ, ಅವಾಗ ಅದು ಫ್ರೀ ಆಗಿ ಇರುತ್ತೆ. ಅಲ್ಲಿ ಕ್ರೋಮೋಜೆನಿಕ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಏನಾಗುತ್ತೆ, ಕಾಲನಿಯಲ್ಲಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಿ ಇಡೀ ಕಾಲನಿ ಬ್ಲೂ ಕಲರ್ ಕಾಣುತ್ತೆ. ಯಾವಾಗ ಬ್ಲೂ ಕಲರ್ ಕಾಣುತ್ತೆ ಅದು ನಾನ್ ರಿಕಾಂಬಿನೆಂಟ್, ಯಾವಾಗ ವೈಟ್ ಕಲರ್ ಇರುತ್ತೆ ಅದು ರಿಕಾಂಬಿನೆಂಟ್. ಸೊ ಐ ಹೋಪ್ ದಟ್ ಫಸ್ಟ್ ಟೆಕ್ನಿಕ್ ಈಸ್ clear. Now look over the second technique. When the foreign DNA is ligated to PAM, H1 site of tetracycline resistant gene of PBR322. So take care of this point. When the foreign DNA is ligated to BAM, H1 site of tetracycline resistant gene of PBR322, it will loses resistance to tetracycline. This can be easily identified from the non-recombinant. It can be easily isolated from the non-recombinant. Look over how they are going to identify recombinant from the non-recombinant. When recombinant and non-recombinant are placed in a plate containing ampicillin, when recombinant and non-recombinant are placed in a plate containing ampicillin, both will grow. When recombinant and non-recombinant are transferred to another plate containing tetracycline antibiotic, only non-recombinant will grow, recombinant will loses their life. Okay, look over, I will repeat this matter. When the foreign DNA is ligated to BAM H1 site of tetracycline resistant gene of PBR322, it will loses resistance to tetracycline. This process is called as insertional inactivation. This can be easily isolated from the non-recombinant. It can be easily isolated from the non-recombinants, or recombinant and non-recombinants can be easily identified by a simple technique. Look over here. When recombinant and non-recombinant are placed in a plate containing ampicillin antibiotic, both will grow. When recombinant and non-recombinant are transferred to a plate containing tetracycline, only non-recombinants will grow, but recombinant will loses their life. Here, ampicillin and tetracycline, one antibiotic will helps to select another antibiotic are both ampicillin and tetracycline plus kanamycin will act as a selectable marker. ನೋಡಿ ಸ್ವಲ್ಪ ಕನ್ನಡದಲ್ಲಿ, ಯಾವಾಗ ನೀವು ಫಾರಿನ್ DNA ತಗೊಂಡು BAMH1 ಸೈಟ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ PBr322ಗೆ ನೀವು ಆ್ಯಡ್ ಮಾಡ್ತಿರೋ ಅಥವಾ ಪೇಸ್ಟ್ ಮಾಡ್ತಿರೋ, ಅವಾಗ PBr322 ಏನು ಮಾಡುತ್ತೆ, ಟೆಟ್ರಾಸೈಕ್ಲಿನಿಗೆ ರೆಸಿಸ್ಟೆಂಟ್ ಕೆಪಾಸಿಟಿಯನ್ನು ಕಳ್ಕೊಂಡ್ಬಿಡುತ್ತೆ. ಈಗ ರಿಕಾಂಬಿನೆಂಟ್ ಮತ್ತು ನಾನ್ ರಿಕಾಂಬಿನೆಂಟ್ ಯಾವ ಥರ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಾರಂದರೆ, ಒಂದು ಪ್ಲೇಟ್ ಇರುತ್ತೆ, ಅದರ ಒಳಗಡೆ ಆ್ಯಂಪಿಸಿಲಿನ್ ಆ್ಯಂಟಿಬಯೋಟಿಕ್ ಹಾಕಿರ್ತಾರೆ. ಅದರೊಳಗಡೆ ನೀವು ರಿಕಾಂಬಿನೆಂಟ್ ಮತ್ತು ನಾನ್ ರಿಕಾಂಬಿನೆಂಟನ್ನು ಬಿಟ್ಟಾಗ ಎರಡು ಬೆಳೀತವೆ. ಆದರೆ ಇನ್ನೊಂದು ಪ್ಲೇಟಲ್ಲಿ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಆ್ಯಂಟಿಬಯೋಟಿಕ್ ಇರುತ್ತೆ, ಅವಾಗ ನೀವು ರಿಕಾಂಬಿನೆಂಟ್ ಮತ್ತು ನಾನ್ ರಿಕಾಂಬಿನೆಂಟನ್ನು ಹಾಕಿದಾಗ ರಿಕಾಂಬಿನೆಂಟ್ ಏನಾಗುತ್ತೆ, ಸತ್ತೋಗ್ಬಿಡುತ್ತೆ. ನಾನ್ ರಿಕಾಂಬಿನೆಂಟ್ಗಳು ಬೆಳೀತವೆ. ನಾನ್ ರಿಕಾಂಬಿನೆಂಟ್ ಯಾಕೆ ಬೆಳೀತಾವಂದರೆ ಅದರ ಒಳಗಡೆ ಇನ್ಯಾಕ್ಟಿವೇಷನ್ ಇರಲ್ಲ ಅಥವಾ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆಂಟ್ ಇರೋದ್ರಿಂದ ಅವು ಗ್ರೋತ್ ಆಗುತ್ತೆ, ಅವು ಮಲ್ಟಿಪ್ಲೈ ಆಗ್ತಾ ಹೋಗ್ತವೆ. ರಿಕಾಂಬಿನೆಂಟ್ ಏನಾಗ್ತವೆ, ಅವು ಡೆತ್ ಆಗಿ ಬಿಡ್ತವೆ. ಓಕೆ, ಲಾಸ್ಟ್ ಪಾಯಿಂಟ್, ಕ್ಲೋನಿಂಗ್ ಸೈಟ್. It should have one or more cloning site or a recognition site. Plasmid should have one or more recognition site. When many recognition sites are present, it will create problem for cloning. So it should have one or few recognition site. When many recognition sites are present, it will create problem for the cloning. So that ends plasmids. So please remember, plasmid should have origin of replication, selectable marker, origin of replication, selectable marker and cloning site. When many cloning sites are present, it will create problem for cloning. For NEET examination or for CET examination, you have to practice this figure. Okay, so where you have to identify the origin of replication, the ampicillin, tetracycline, and here there are 8 restriction endonucleases sites they have mentioned: PST1, PVU1, ECOR1. Already we have come across E. coli, R strain, Roman letter 1, CLA1, HIND3. Previously we have come across HIND2, Haemophilus influenzae, D strain, Roman letter 2. Here you have to remember HIND3. BAMH1 site, SAL1, PVU2. Okay, you have to take care of all these labeling parts. Okay, dear students, now look over The next matter, vectors for cloning genes, vectors for cloning genes in plants and animals. Vectors for cloning genes, vectors for cloning genes in plants and animals. 2 points are there, you have to take care. Number 1, retroviruses. Retroviruses have the capacity or their capability of converting, retroviruses are capable or capability of converting the normal cell into cancerous cells. Retroviruses are capable of converting or they have capability of converting the normal animal cell into cancerous cell. I hope that you know cancer. Cancer is an uncontrolled division of the cells leading to the formation of tumors. Retroviruses have the capability to convert the normal animal cell into cancerous cell. Cancer is an uncontrolled division of the cells. Cancer is an uncontrolled division of the cells leading to the formation of tumor. Cancer is an uncontrolled division of the cells leading to the formation of tumor. Now these retroviruses are altered so that they will become non-pathogenic. Retroviruses are altered. Now they are non-pathogenic and are used to transfer the desired genes in animal cell. Look over, retroviruses are having capability to convert the normal cells, normal animal cell into cancerous cells. Now retroviruses are altered so that they are non-pathogenic and they have the capacity to transfer the desired DNA in animal cell. Same way, Agrobacterium tumefaciens, Agrobacterium tumefaciens are the soil bacteria which has got the capability of converting the normal cells into cancerous cells in especially dicot plants. Look over here, Agrobacterium tumefaciens are the soil bacteria. It is having tumor inducing plasmid. It is having tumor inducing plasmid with tDNA. When tumor inducing plasmid with tDNA is transferred to a normal plant cell, it will convert the cell into cancerous cell. Plant cancer is commonly called as crown gall. Crown gall is nothing but a plant cancer. Appearance of the gall on the crown of, appearance of the gall on the crown of young fruiting trees and herbaceous plant is called as crown gall. Appearance of the gall on the crown of, appearance of the gall on the crown of young fruiting trees and herbaceous plant is called as crown gall. Generally, these crown galls are noticed in the herbaceous plant and even fruit yielding dicot plants. Agrobacterium tumefaciens are the soil bacteria. It is having tumor inducing plasmid with tDNA. When this tumor inducing plasmid with tDNA is incorporated into the normal cell, it will convert it into cancerous cell. Now agrobacteria is altered. Okay, it is altered and it is non-pathogenic and are used to transfer the desired DNA in the plant cells. So that ends vectors for cloning genes in plants and animals. Okay, I hope that agrobacteria are used to transfer the desired genes in plant. Retroviruses are used to transfer the desired genes in the animal cell. Now both are altered and they are non-pathogenic. Now look over, dear students, competent host. DNA, DNA cannot easily pass through the cell membrane. DNA cannot easily pass through the cell membrane because of hydrophilic nature. So this word you have come across in PUC first year chapter transport in plants. The substance which has got a great affinity towards water is called as hydrophilic substance. The substance which has got a great affinity towards water is called as hydrophilic substance. Okay, so look over here, competent host. DNA cannot easily pass through the cell membrane because of hydrophilic nature. Now we have to make the host cell. Now we have to make the host cell as an competent by treating them with a cold calcium chloride solution. Cold calcium chloride solution. Followed by that, we have to go for heat shock for 42 degrees centigrade. I will clear this matter. DNA cannot easily pass through the cell membrane because of hydrophilic nature. The substance which has got a great affinity towards water is called as hydrophilic substance. DNA cannot easily pass through the cell membrane because of hydrophilic nature. So you have to make the host cell as an competent. You have to make the host cell as an competent by treating them with cold calcium chloride solution. Followed by that, you have to go for heat shock for about 42 degrees centigrade. Brief heat shock for 42 degrees centigrade. Followed by that, once again the cell is placed in ice. Once again the cell is placed in ice. This will destabilizes the plasma membrane and helps in making the pore. That helps the cells to take recombinant DNA. Look over here. DNA cannot easily pass through the cell membrane because of hydrophilic nature. So you have to make the host cell as an competent by treating them with a cold calcium chloride solution. You know already calcium is a divalent ion. Followed by that, you have to go for brief heat shock for about 42 degrees centigrade. Then once again it is placed in ice. That will destabilizes the plasma membrane and helps in making the pore. That will take, or the host cell will take recombinant DNA. Okay. Second technique, micro injection, where the recombinant DNA or foreign DNA is directly introduced into the nucleus of an animal cell with the help of micro pipettes. In micro injection, recombinant DNA or desired genes are directly introduced into the nucleus of animal cell with the help of micro pipettes or micro syringes. Pipettes you have come across in chemistry laboratory. Micro syringes are the small sized syringes. Okay. Next, third is electroporation, where making the cell porous by electric current. In electroporation, cell is made porous by electric current. Cell is made porous by electric currents. Last one, gene gun or biolistic, where the plant cell is bombarded with high velocity microparticles. Microparticles means it measures around nearly 1 to 2 mm in size. Look over the last technology, gene gun or biolistic, where the plant cell is bombarded with high velocity microparticles of gold or tungsten coated with DNA. Okay, so in microinjection, recombinant DNA or desired DNA or foreign DNA is directly introduced into the nucleus of an animal cell with the help of micropipette or micro syringes. In electroporation, Pore size of the membrane are making the cell porous by electric current. In gene gun or bioelastic, plant cell is bombarded with high velocity microparticles of gold or tungsten coated with DNA. Size of that gold or tungsten is 1.2 mu m. Okay, so that ends 4 technologies to make the competent host cell. Even disarmed pathogens are also used as an vector. Okay, disarmed pathogens are also used as an vector.